Good morning, children. Welcome to biology class. One of the major experiment for our practical examination is DNA isolation. So we now completed the lesson Biotechnology Principles and Processes. There we explained the steps of rDNA technology. So the first step is DNA isolation, isn't it? We discussed already about the isolation of DNA. Today, I will demonstrate that experiment for you. So, the material required for the experiment are liquid soap, salt, water, then methylated spirit or any plant material. Okay? And keep it in mind, this ethanol should keep in refrigerator 24 hours. Okay? It should be chilled ethanol. Okay, it is very important. Any plant material can use for this experiment. Today we are selecting the banana. So the first step is crushing of plant material. So the principle behind this is this crushing degrades the cell walls and cell membrane, further permitting the release of DNA. But do not blend for too long as this will break up the DNA fibers. Then the second step is we are adding salt soap solution. We are using both salt and soap and make it a solution. And why we are adding detergent? The detergent and heat treatment degrades membrane phospholipids and proteins releasing the DNA. And what is the use of salt? The positively charged sodium ions from the salt shield the negatively charged phosphate group of the DNA molecules, helping them to precipitate out of solution. Okay. Then we allow to cool it. This slows the breakdown of DNA, which would occur if a high temperature was maintained. Then in the last step, we are adding ethanol, and keep it in mind, we should add chilled ethanol. And what is the principle behind that? It will precipitate into upper layer of ethanol, and the DNA is a white material and the clear alcohol layer. This step, and the first step, we are adding detergent and soap solution, so it helps to disrupt the plasma membrane. Okay. Then we are filtering the solution, and into that filtrate we are adding chilled ethanol. It helps to precipitate the DNA out. And you can see before and adding ethanol, see the difference. So after adding the chilled ethanol, you can see this precipitated DNA. Can you see the precipitated DNA? Okay. So let's start the experiment. Materials required for this experiment are soap solution, salt, then ethanol, distilled water, and a plant material. Here I am taking a banana, then a Ziploc bag. Okay. So by using soap solution and salt, we have to prepare a solution. I prepared that. Then the first step is take a small piece of banana and keep it in the Ziploc bag. Then make a smash. Close the bag. Then make a fine smash. Then add this soap salt solution to it. Again close the bag. Then the solution filter by using a filter paper. Then wait for some time. Then take the filtrate. Then the last step is adding ethanol. Children, keep it in mind, we should add chilled ethanol. Otherwise, the DNA can't precipitate out. Okay? And add this chilled ethanol slowly through the side of the test tube. Then wait for some time. See? You can see the precipitated DNA in the test tube. Can you see this? Thread shaped, white colored DNA. Then using a glass rod, take the precipitated DNA out and keep it on center of your slide. Okay, and submit it to the examiner. So, the experiment isolation of DNA is completed. Very simple experiment, isn't it? Only 3 steps are there. What are the steps? First of all, we take a plant material. So here I take a piece of banana. Then add soap and salt solution to it. Then make a smash. Then filter it using a filter paper and take out the filtrate. Then add chilled ethanol. So still it is there. See the test tube. In the supernatant you can see fork like precipitation. Can you see it? Then by using a test tube, take out it and keep it on the slide. So this is the experiment isolation of DNA. Okay children?